Hello, and welcome to Arcadia University's University Seminar 275 Scientific Ethics. I'm John Hoffman, and this video is going to give us an overview of Unit 6, which doesn't have a great name because I couldn't come up with one, but we're going to call it Genetic Ethics, or the Ethics of DNA, or the Ethics of DNA Technology, but in essence, Genetic Ethics. And so what we're going to do within this unit is essentially have a series of lectures. We're going to talk about DNA and how DNA can be used within the treatment of diseases as well as the treatment of a range of conditions. So to start out with, again, what we need to do is have a better understanding about what is the DNA molecule and what type of information is it's going to be carrying. And so we'll start out with some of the science. We're going to talk about the basic structure of DNA and its relationship with chromosomes. We're going to talk about how the message of DNA is coded into a gene and identify and define some of these scientific terms as we're going through. And then talk about how genetic information is going to be passed from parent to offspring. Again, recognizing that an individual a son or a daughter is going to have similar characteristics that they're going to be receiving from their parents. The topic, again, like we've had throughout this course, we'll have a series of discussion topics that you'll be doing with your discussion group. And we're going to talk about the inheritance of traits and essentially taking a look at whether it's ethical to have a child if you know that there's a chance that they're going to have some type of severe genetic condition. And think about some of the ethical considerations that are involved. Think about what are the odds. Think about what types of things need to be thought of as we're going through and taking a look at the transmission of characteristics, in this case the transmission of a debilitating genetic condition from a parent to an offspring. The next thing we're going to look at is going to be DNA testing. And this is kind of a crude way of looking at it. But in order to understand what can be used for DNA, what can be used with DNA, one of the ways in which we can do is take a look at DNA patterns and studying these DNA patterns in order to be used for identification. So looking at paternity testing, looking at the CODIS system that the FBI uses for identifying potential criminals associated with a crime. But to take a step further and say, okay, should we have a genetic census? Should we have a situation where the genetic characteristics of every individual is essentially housed in some type of database at some location? And the topic associated with this kind of follows up from that. When should information on a genetic census be used? We know that we have the ability to do paternity testing, but we have to essentially obtain the DNA from the child and the potential father to test it. We could do DNA testing to identify criminals, but it's dependent upon their DNA being in the database, so they need to have been arrested for something previous. But should we have it to identify criminals that haven't been arrested for something before? Should we use this as a data bank for identifying potential matches for bone marrow or blood transfusion or organ transplants that we've talked about previously within the course? What limits should be used in determining whether genetic census should be created, and if it's created, how can that information be used in an ethical and appropriate way? Next, we're going to look at DNA analysis, and the difference between testing and analysis is pretty slight, but I'm going to claim that the testing is doing like the identification. The analysis is actually going through and looking at the specific sequence of the genes, and we'll talk about that in a little bit more detail, but essentially understanding the specific characteristics instead of the patterns used for identification, the specific information within an individual's DNA that can be used to figure out whether somebody is at risk for a disease or whether somebody has a characteristic that gives them a benefit or is a detriment in some way. So understanding how we get to the very specific details within the DNA, what can be learned about what's possible for that individual. And related to that is this concept of gene patenting. People that are scientists or companies that are studying the very specific information with the genes, in many cases, have gone through and filed a patent for that gene. And so, what is, in essence, a set of information that's found within a human body, within a human being, is now patented, and because it's patented, it's restricted, or at least the research on it, the use of that information, is controlled by some entity, by some company, in essence, that holds that patent. And so we need to think about when is it ethical, or is it ethical to patent genes, patent the information that's found within the human genome, within an individual's human DNA. Then we're going to start to say, okay, once we understand what's in the DNA, we're going to get a little bit more technical. We're going to say, okay, once we understand it, once we know what's there, let's go through and start to change it a bit. And so we're going to look at recombinant DNA, which is in essence a way of going through and modifying that DNA in some way. Generally, it means mixing DNA from two different sources into what's called a recombinant or recombined molecule. But the potential is there for us to go through and kind of customize the information that's found within a DNA molecule. Lots of possibilities, but lots of ethical concerns associated with it. And so what we're going to look at is, when is it possible, when is it permissible to use this genetic engineering, this recombination, to create strains of bacteria to do specific functions. We'll talk about some specific examples that are used right now, but recognize that bacteria and yeast have been used for years in the production of bread and wine and cheeses. Now we have the potential to modify these bacteria, modify the yeast if we want to do that, and get it to do specific things. So we can get it to a state where a bacterial cell will produce a human drug, like a human insulin that can be used for the treatment of diabetes, or a human clotting factor that can be used to treat an individual that has hemophilia. Essentially, they don't have the ability to stop bleeding, in essence, when they have a cut or an injury. When is it appropriate? What are the limits on what we can do in modifying these bacteria for human use? Then going from bacteria to more complicated organisms. So in essence, the bacteria are still going to be genetically modified organisms, but we're looking at kind of larger organisms, organisms that we can see and think about and we're interacting with. And so in terms of genetically modified organisms, we're going to take a look at how we use that technology, that recombinant DNA technology, to go through and change the genome, to go then and change the characteristics of an organism. And we're going to talk about that in a variety of examples where we're looking at genetically modified crops that have been modified in some way to address a need, and then the concerns associated with it. And the discussion topic for this is going to be taking a look at golden rice. It's a genetically modified crop that was created to increase the nutritional properties of a rice, but for years, I think we're up to almost 15 years now, that it's been possible to do this, but it still hasn't been approved for widespread use. And so we talk about what type of regulatory criteria should be present in order to be able to determine when a genetically modified material possibly could, or whether or not it should or should not be used for human consumption. And again, talk about this, the science involved, as well as the ethical considerations, the safety considerations. And then finally, we're going to talk about gene therapy. And so we talked about it as genetically modifying bacteria. We've talked about genetically modifying organisms and put the example as modifying crop plants. But we also have the ability to go through and genetically modify a human being. And so this concept of gene therapy is to use this concept of gene recombination, DNA recombination, this DNA technology in essence, for the treatment of a human disease. And we can do this in two different ways. We can treat it within the somatic or body cells of an individual, and treat the disease within that individual, or we can treat it in the germline. And now if we treat it in the germline, treat it in either the egg or the sperm, we essentially make it hereditable. We make it so whatever change we made can be passed on to future offspring. Now it's nice because if we do this and we create the cure in essence, all future generations will be cured of that disease. But there are incredibly controversial ethics associated with that. Big fears, big concerns, lots of things that need to be considered before we get to that point. Now, with gene therapy, we're getting down to the end of the course, and there are a couple kind of topic questions that you can take a look at, but there's no assignment because I want you to essentially finish up the final stages of your project. And so that's the overview for this last concept, the genetic ethics, DNA ethics, within the course. And so if you have any questions, feel free to email me at hoffmanj@arcadia.edu. Thanks.